Hello students, in this lecture we are going to discuss about the next primary metabolite, that is amino acids and vitamins. What are amino acids? Here the general structure of amino acid has been shown, which consists of the amino group, that is NH2, the carbon, to that carbon amino group is been attached. Next is carboxyl group, that is COOH, then hydrogen, and any alkyl group or any side chain is attached, that is indicated by the capital R. So this is the general structure of amino acid. Here amino group, carboxyl group and hydrogen, these three remain constant, and just the side chain, that is the R group, is been exchanged or has been replaced by different structures, and then the particular amino acid is been formed. Okay, so what are amino acids? Amino acids, these are the building blocks of the protein. Means protein is formed by the amino acids. Then this protein may be in the form of primary structure, secondary structure, tertiary structure, depend. Okay, so that are the building block. They consist of the amino group and carboxyl group. Amino group is basic in nature, while carboxyl group is acidic in nature, as we have shown the general structure of amino acid, and this R group is specific to each amino acid. There are different examples of amino acids which are being produced at the industrial level, like lysine is there, glutamic acid is there, methionine is there, etc. Various amino acids are being there. Then these amino acids are being grouped into different types, like aromatic amino acid, acidic amino acid, basic amino acid, polar amino acid, non-polar amino acids. So that is the different theme of the amino acid, but our main criteria is to focus on the production of the amino acid, the organisms which are required for the production of the amino acid. That points we are going to focus in this lecture. So the lysine, the main amino acid that we are going to discuss in this is the lysine production. Okay, so lysine production, the fermentation has been carried out by submerged fermentation or aerobic fermentation. Mode of operations are batch process, as batch fermentation we have discussed in our previous video, fed batch process, and the fermenter type stir tank reactors are there, that is CSTR, continuous stir tank reactor. Even that point also we have discussed in our previous video. And the next type of the fermentation fermenter is the airlift bioreactor. This is the different type of bioreactor, like means how CSTR is there. In the same case, airlift bioreactors are there. So let's start with the production of the lysine. The microorganism responsible for the production of the lysine are Corynebacterium glutamicum. This organism or this bacteria, it is widely used for the production of different amino acids, like glutamic acid. Glutamic acid also means glutamic acid is also been produced by the Corynebacterium glutamicum, and lysine is also been produced by the same bacteria. Corynebacterium glutamicum, if we will see, this bacteria is gram positive, rod shaped, non-motile, non-spore forming, non-pathogenic, and it is easily found in the soil. Okay, so it is the soil bacterium. Now lysine production, if we will, now let's start with the production. When you discuss about the organism, from that point only we have started our production. The next point is the media composition required for the lysine production. So media, carbon source include the cane molasses. This is the viscous product obtained from sugar cane. When sugar cane is been processed, or when sugar cane is been refined, the extract, which is the viscous product which is obtained, that is known as the molasses. So it acts as the carbon source. Next is the nitrogen source. Corn strip liquor you can use, or soybean meal you can use. Now what is corn strip liquor? Corn strip liquor, it is the byproduct of corn wet milling. Okay, it is the just byproduct of corn, and which is very rich in protein, rich in energy, rich in minerals. So it has the high protein and high energy content. Next, mineral salts you can use like KH2PO4, K2HPO4, calcium carbonate. Even this minerals and salt, it maintains the pH of the fermenter media. Then trace elements that are required in very minute amount. So in the trace elements also you can use the same as of the nitrogen source, that is Const-Replica. Antifoam agents you can use like PEG 2000 and silicon based oil. PG stands for polyethyl glycol. Okay, so this can be used as the anti-foam agent to prevent the foam formation. Now the procedure, even apart from the cane molasses, like means carbon source, which is the cane molasses, apart from that you can use glucose, fructose, sucrose and starch also. And in nitrogen source the Const-Replica is been said. Apart from that you can also use ammonium nitrate salt and yeast extract. So yes, the optional mode also you can use. The pH of this culture condition is 7 and it is been neutral temperature, which is 28 to 30 degrees Celsius. And for the production of lysine, high oxygen concentration is been required. Okay, so this is all about the we can say fermentation media and the cultural condition. The process has been carried out by the three steps, that is fermentation, product recovery, product concentration, drying, and then final is packaging. Okay, so let's start with the fermentation media. Now fermentation media, the fermentation is been carried out using the batch type of mode. Means as we have discussed about the batch fermentation, nutrients are being added, final there is product formation, the first batch is completed, the fermenter is been cleaned, it has been sterilized, and then second batch is been processed. Okay, so actually what happens in the production, the organism along with this media, carbon source, nitrogen source, and etc etc what we have provided, the organism is been inoculated and then fermentation process is been run out. Now actually what happens in the fermentation, that we will see. Now lysine biosynthesis is carried out in two pathways. It is not mentioned in the slide, so listen it properly. It is carried out in two pathways. First is DAP pathway and second is AAA pathway, that is AAA pathway. We will discuss first about the DAP pathway. DAP stands for diaminopimelic acid. Okay, diaminopimelic acid. It is an intermediate which is found in the bacteria, algae and higher plants. Okay, DAP is found in the bacteria, algae and higher plants. So pyruvate is first converted into the aspartate and then DAP is formed and then DAP is finally converted into the lysine. So this is overall pathway. Means we have said that we have discussed that lysine is been formed, but what reaction is been carried out in the fermenter? So this one of the reaction is there, that is DAP pathway, which is the intermediate product formed in this pathway. Okay, and it is generally found in the bacteria, algae and higher plants. So pyruvate we have add the carbon source in the form means molasses and glucose or fructose we have been added. That glucose is been converted into the pyruvate plus aspartate is the amino acid, and then this aspartate by the intermediate product it has been converted into DAP, and then DAP finally convert the substrate into the lysine product. So this pathway actually occur in the bacteria known as the Corynebacterium glutamicum, which leads to the production of the lysine. Second pathway is AAA pathway. AAA stands for alpha amino adipate. Okay, so alpha amino adipate, it is also an intermediate found in the fungi. The reaction steps are acetate plus alpha ketoglutarate. It converts acetate plus alpha ketoglutarate into AAA. It means the bacteria converts into AAA, and then finally there is production of the lysine. And animals do not synthesize the lysine. It is essential amino acid. I forget to tell that there are two classes of amino acid: essential amino acid and non-essential amino acids. So what are essential amino acid? Essential amino acids means which cannot be synthesized by the body. Okay, so therefore it has to be supplied through the external source or through the diet. So that are the essential amino acids, and non-essential amino acids are the opposite of essential amino acids, means they can be synthesized by the body, so there is no need to provide externally. So there are two classes of the amino acids. So yes, this is the two pathway which leads to the production of the amino acid, and different vitamins and antibiotics also stimulate the growth and lysine yield. Apart from the source, carbon, nitrogen and these sources, we can add externally vitamins also and some antibiotics also. Why? To stimulate the growth and to increase the yield. Yield means finally production what we have got, okay, quantity. So to increase the yield of the lysine, we can use these vitamins and antibiotics. So this was overall production of the lysine. If we will see the product recovery, means finally the fermentation process has been done and you have your product, so you have to purify your product, so you will go for the product recovery, or we can say downstream processing. तो product recovery ये की जाती है by filtration, ultra filtration किया जाता है, means you filter your desired product from the media, and then it is also done to the ion exchange column. अभी ion exchange column क्या है? It is the technique of chromatography. Okay, so it is the chromatography technique. मतलब एक कॉलम क्रिएट किया जाता है, एक जार में कॉलम क्रिएट किया जाता है, और उस कॉलम से your lysine has been flowed. Okay, so it attached to that it attached to the resin, और फिर आफ्टर दैट मोबाइल ऊपर से जो ऊपर से जो मोबाइल फेज कैरी आउट की जाती है, तो मोबाइल फेज जो फ्लो की जाती है अलॉन्ग विद द स्टेशनरी फेज, उसके थ्रू आपका लाइसिन सेपरेट आउट होता है. सो रिकवरी बहुत डिटेल में प्रोसेस है, एंड इट्स नॉट the recovery process is not been important. Just keep in mind that the recovery is done by the ultrafiltration technique and by using the ion exchange chromatography, and then finally in the dry form the product has been recovered and it is been packed. If we will see the applications of this amino acid lysine, then it is used as the nutrient supplements in food, beverages, pharmaceutical industries, and it also helps to improve the nervous system. So this is all about the lysine, its production, and how it's been produced, or what are the applications of this lysine. Let's start with our next, that is vitamin. Now we will see about the vitamins. Vitamins are soluble. There are two types of vitamins: water soluble vitamins and fat soluble vitamins. Before going to this type, we will see what are vitamins. So vitamins, these are required for normal functioning of our body system. Okay, so that are the vitamins. These are the organic molecules. So two types are there: organic molecule and essential micronutrients. Vitamin, this word has came from the Latin word vita. Vita, vita means life. Okay, so that is the organic essential micronutrient. Two types are there: fat soluble, water soluble. So fat soluble vitamins are stored in the body fatty tissues. Examples are vitamin A, D, E and K. And water soluble vitamins, they dissolve in water easily. As the name indicates, water soluble means these vitamins are soluble in water, they dissolve in water. The examples are B complex vitamin and vitamin C, means all vitamin B1, vitamin B2, riboflavin, then thiamine, pantothenic acid. मतलब ये जो vitamin B1, B2, B3, B4 है, उसी के ये scientific names है. Okay, respectively. So this was all about general introduction about of vitamin, and vitamins are required in we can say a small quantity to carry out the normal functioning of our body. In this video we will discuss about the production of vitamin B12. Scientifically it is known as the cyanocobalamin. Okay, so cyanocobalamin you can say, or just cobalamin भी आप उसे बोल सकते हैं. तो कोबालामाइन क्या है? कोबालामाइन सबसे पहले इट इज़ द water soluble vitamin which is important in normal functioning of brain and nervous system. Plus it also plays main role in the formation of red blood cells, that is RBCs. Okay, it is not found in nature, naturally वो find नहीं होता है, it has to be produced at industrial level, and then it is being sold in the form of the tablets, or yes, it's sold in the form of the tablets. So it is carried out by the submerged culture process, and it requires overall fermentation of 3 to 5 days. The microorganism which is used in the production of this vitamin B12 is Streptomyces gracius, Streptomyces olaviceus, Bacillus megaterium, Bacillus coagulans, Pseudomonas denitrificans, Propionibacterium friedenrichi, Propionibacterium shermani, and a mixed fermentation of a Proteus species and Pseudomonas species. So see, there are hundreds, means there are a large number of microorganisms involved in the production of the vitamin B12. Okay, so that is been there. Mostly Streptomyces gracius and Streptomyces olaviceus is been used for the production of the vitamin B12. Now, what are the steps in the production? The steps are similar to that of the production we have shown previously. Same: formulation of media, sterilization of medium, making starter culture, then anaerobic fermentation, aerobic fermentation, and finally recovery. या just additional anaerobic fermentation पहले किया जाता है and then aerobic fermentation किया जाता है. Okay, so first step in that, the main thing is preparation of inoculum. Now, how you will prepare the inoculum? What is inoculum? That we have discussed in our previous lectures. So here, pure culture of Streptomyces. Now we have to prepare vitamin B12 by using the species Streptomyces olivaceus. Okay, so pure culture of this olivaceus is inoculated in 100 to 250 ml of inoculum medium. Okay, जो slant होते हैं Streptomyces olivaceus के, उनका कल्चर इनोकुलेट किया जाता है इनटू दी 100 to 250 ml ऑफ इनोकुलम मीडियम, एंड देन दिस फ्लास्क इज इंक्यूबेटेड ऑनटू दी शेकर टू प्रोवाइड द एरिएशन. ओके, शेकर में इंक्यूबेट किया जाता है, सो इट कॉन्स्टेंटली रोटेट्स एंड प्रोवाइड द एरिएशन. सो दैट इज योर एरोबिक टाइप ऑफ वी कैन से इंक्यूबेशन. ओके, and then this flask culture is subsequently used to inoculate into the larger tanks. Okay, जब उसकी ग्रोथ हो जाती है तब ये इनोकुलम ऐड किया जाता है into the larger tanks. Then media used for the production of the this vitamin B12 is yeast extract यूज़ किया जाता है, beef extract यूज़ किया जाता है, glucose, agar, and the pH which is maintained is about 7.3. सो दैट इज़ बीन देयर. अगर आप बैच टाइप ऑफ फर्मेंटेशन यूज़ करते तो उसका टेम्परेचर है 250 डिग्री फ़ारेनहाइट फॉर 1 hour. इफ़ यू यूज़ कंटिन्यूस टाइप ऑफ fermentation, then the temperature is 330 degree Fahrenheit for 13 minutes by mixing with the steam or by mixing with the vapor. ठीक है. एडिशनली ये जो बीफ एक्सट्रैक्ट है, ग्लूकोज एक्सट्रैक्ट है, आगार है, ओके, उसके अलावा एडिशनली यू कैन ऐड डेक्सट्रोज कैल्शियम कार्बोनेट, प्लस ग्रोथ स्टिमुलेट करने के लिए आप डाल सकते हैं कोबाल्ट, आयरन एंड अदर सॉल्ट. ओके, सो इट इंक्रीजेज द यील्ड एंड स्टिमुलेट्स द प्रोडक्शन ऑफ विटामिन B12. सो दैट इज द प्रोडक्शन मीडियम. टेम्परेचर, पीएच, एरिएशन, एजिटेशन, एंटीफोम एजेंट, ये सब आप टू प्रिवेंट द कंटामिनेशन और टू मेंटेन द मॉनिटरिंग ऑफ दैट मीडिया यू हैव टू कंट्रोल ऑल दिस पैरामीटर्स. टेम्परेचर इज मेंटेन एट 80 डिग्री फ़ारेनहाइट. पीएच होता है स्टार्टिंग में पीएच मीन्स एट स्टार्टिंग ऑफ द प्रोसेस पीएच फॉल्स. ओके, पीएच डाउन होता है ड्यू टू रैपिड कंजम्पशन ऑफ द शुगर, बिकॉज शुगर ऑर्गेनिज्म यूटिलाइज करने लगते हैं. ठीक है, सो पीएच उसका फॉल होता है एंड देन इट राइजेस आफ्टर 2 to 4 ड्यू टू लाइसिस ऑफ दैट माइसेलियम. ओके, सो पीएच 5 मेंटेन किया जाता है with the addition of H2SO4 or Na2SO4. Then antifoam agents are being used. Antifoam agents like soybean oil, corn oil, So yes, these are the parameters which are very common for every production. एंड नेक्स्ट स्टेप प्रोडक्शन मीडियम की अगर हम बात की जाए तो प्रोडक्शन मीडियम इट कंसिस्ट ऑफ द कार्बोहाइड्रेट प्रोटीनेशियस material and source of cobalt and other salt. Why it is necessary to add cobalt into the medium? Just to get the maximum yield of the cobalamin. Okay, so we add the cobalt. So cobalt, it is been added, is the compulsory source. Then cyanide is added for the conversion of other cobalamines to the vitamin B12. इट इज़ द बायोसिंथेटिक पाथवे. साइनाइड ऊपर से ऐड किया जाता है कोबाल अमाइन का कन्वर्जन फाइनली विटामिन B12 में करने के लिए. एंड देन नेक्स्ट इज द रिकवरी प्रोसेस. द रिकवरी प्रोसेस इज डन इन द सेम मैनर बाय फिल्ट्रेशन और बाय द अल्ट्रा फिल्ट्रेशन, बट हियर the pH is very important. It is maintained at the pH 5. Then why filtration is being done? To remove the mycelium, or just to obtain our vitamin in the form of the powder, or in the form of the crystals. Okay, so this is the production about the vitamin B12 and amino acid. So in next video we will discuss about our next production. Thank you.